Good morning. This is Michael Dong, and here is episode number 10 on bonded phases and also the Van Deemter equation. Here's the agenda on episode number 10. We will talk about bonded phase types, bonding chemistry, and the Van Deemter plot. So here is the three different sections. First one is types of bonded phase and selectivity differences. We discussed bonded phases for reverse phase, normal phase, ion exchange, and selectivity differences for reverse phase bonded phases. Number two, bonding chemistry. We talk about traditional bonding chemistry using monofunctional silanes, the process of end capping, and the importance of residual solenols, and the need for something called high purity silica. Number three, we discussed the Van Deemter equation. What is plate count and what is the three Van Deemter equation, A term, B term, and C terms, and their ramifications, and how to estimate plate counts from the length and also the particle size. Here's a slide on the common bonding chemistry, starting with reverse phase. The most important, obviously, is C18 octadecyl silane, uh, which is used in most HPLC application. Estimate of anywhere from 60 to 80% of application using reverse phase using C18. Here's C8 and cyanol, phenol, something called polar embedded phases, which are really important for selectivity, uh, fine tuning in method development. This particular one using an embedded amide group in the hydrophobic chain. So in the middle of the chain, there's a embedded amide group. For normal phase chromatography, silica can be used. It's not the best because of the chromatography is not as consistent due to contaminations. If you do a bonding of amino group onto silica surfaces, it make the normal phase less susceptible to contamination. Still, it's not the best. And then we have ion exchange. We have cation exchange is also anion exchange in the last two. Okay, here we discuss the traditional bonding chemistry using monofunctional silane. The example we use is using a Ca bond phases. So some of the hydrophobic chain is Ca instead of C18. And here's a schematic of the silica surfaces with free solenols. Uh, Granted that most of the solenols, because these are spherical porous silica, are inside of the silica particle in the interstitial volume inside. And here's our bonding reagent, which is octyl monochloro octyl dimethylsilane. So it's kind of a functional silane with the chlorine group as a leaving group, the chlorides as a leaving group after condensation reaction, bonding with the silicon material. Now, in order to reduce residual cyanide group, because the fact that most of the cyanide groups are buried inside, sometimes because of the bulky group of the C18 group, not all the cyanide group are accessible, leaving a substantial number of residual cyanide groups uh, in the surfaces. In order to reduce the number and also the activity of that, we use an end capping reagent. And most bond phases has been end capped to reduce the number and the activity of the residual cyanide group. And typically we use a smaller group such as chlorotrimethylsilane in order to assess some of the more difficult to assess pores uh, and make sure that as many residual cyanide are bonded. So monofunctional silane is a traditional bonding chemistry, and end capping is used in most phases to reduce the number of residual cyanide groups. Here's another look at the schematic of the silica before bonding and after bonding, right? So uh, pretty similar to what we saw in the previous slide. And you see the surfaces uh, uh, on the silica backbone support, and after bonding with the Ca material, there are residual cyanide groups remaining behind. So even after extensive bonding and end capping processes, still there's incomplete coverage since most cyanodes are inside the pore. So there's still a large number of residual cyanide groups. And these can cause peak tailing for basic analyte. For any pharmaceutical compound, which tend to be mostly basic analyte, weak basis, it could be a problem for peak tailing. The number of residual cyanide group and the acidity or the activity of cyanide group is very important, and it actually causes batch-to-batch inconsistency or non-reproducibility in the bonding phases. So it was a big problem uh, prior to 2 decades ago in the 70s, 80s, and even 90s that these residual cyanide group, due to activated residual cyanide group, cause batch-to-batch problems. Okay, in the late 90s, research from Alois Snyder and Jack Kirkland, they looked into the different type of cyanide group, and they found out that cyanodes are not created equal, meaning that the activity are not the same depending on how associated the position of the cyanide group. So you have associated cyanide group which are hydrogen bonded to each other, they are adjacent, and therefore they are pretty non-active. The germinal cyanide group are more active, they're increasingly more acidic, and free cyanide are actually the most acidic. However, it turned out that the most acidic cyanide group are those which are adjacent to a metallic oxide. It turned out that in the 70s and 80s, uh, a lot of the silica material are coming from minerals which are mined and then sift to give you the right size. So they are not synthesized. So the purity is not good, even though that the particles are very strong with metallic oxide. Now it turned out that metallic oxide in the silica material cause uh, the cyanide group to be highly acidic because that the lone pair of oxygen on the oxygen group can be delocalized into the empty orbital, the empty d orbital of the metallic atoms. So so that actually became a really important issue. So in order to reduce the activity, uh, you know, after this pivotal discovery, the cure is to actually make a silica which is high purity. And one of the first high purity silica material, Boniface, is called water symmetry in the early 1990s. Okay, and here is a comparative chromatogram of a 5-component test mixture. Two of these are basic component, and we're comparing water symmetry, which is the first generation of high purity silica material making into a Boniface versus hyposil BDS. And here we not the mobile phase is pretty straightforward, without any ionic salt or additive right here, 75% methanol water. And we're comparing the chromatogram and the performance in terms of peak shape. And you find out that while the efficiency are very similar for the neutral compound, you can see that for hyposil BDS, The peak shade of 2 and 4 are just terrible. They're highly tailing. And this is because this particular hypercell was not made from high purity silica. Therefore, there's substantial amount of metallic oxide in the bulk silica, causing the residual cyanode to be highly acidic. And therefore, any basic analyte can have acid-base interaction, um, causing the peak to be tailing substantially. In this section, we talk about novel bonding chemistry and some of the newer processing other than just monofunctional silane. So there's some improvement in more efficient bonding. So traditional bonding using monofunctional silane uh, typically achieve only about 50% efficiency, or about 2 micromole per meter square of bonding, meaning there's a substantial amount of residual cyanide group in there. And there's a increasing tendency to use something called polymeric bonding chemistry using polyfunctional silane, mostly trifunctional silane, meaning that instead of monochloro silane, use a trichlorosilane. In the beginning, people also used difunctional silane. It turned out not to be very reproducible. So I think that chemistry has been abandoned. So many modern bonding bonded phases employ trifunctional silane, such as case of water security C18. There are other bonded phases, such as the one called phenol, and this one is a pentafluorophenol, which is very effective for analyzing more polar drugs. Uh, there's also a class called the polar embedded group, in which one actually bond a polar group within the hydrophobic chain. So it give the bonder phase a mixed mode interaction. So this is actually a very effective bonder phases to give you a mixed mode interaction, and great for method development when C18 does not work in separating all the critical pairs of analytes. So in terms of retention mechanism, CA and C18 are mostly using hydrophobic interaction, right? Very simple dispersive forces as the retention mechanism. Phenol uh, have some can have pi-pi interaction because of the phenol. So since most drugs have aromatic residue in the drug structure, this would give this monophase different selectivity in HPLC impurity testing methods. We have PFP, which give you pi-pi interaction, dipole, and also hydrogen bonding, because fluorine atoms are highly electronegative. A polar embedded group also had hydrophobic, dipole interaction, and also hydrogen bonding. There are some novel bonding chemistry shown here. Uh, there's a patented technology called a stable bond, which eventually became Agilent's Sorbax. It was developed and invented by AMT from Dr. Jack Kirkland. So the way they do the bonding is instead using a methyl group, such as here, the silane bonding chemistry is actually in isopropyl group, and this is actually designed so that to shield the effect of the residual cyanide group on that, right? So to make it kind of bulky on the side, so that the analyte is less able to assess the residual cyanide group. Typically, for stable bond, we do not do any end capping. Okay. Also, in the late 90s, there's also the development of these phases, which is difunctional bonding chemistry, where one used a trichlorosilane. And this became the exterior mass spec bond phases, and also the one of the first embedded polar group. In the water's chemistry, the nomenclature is called the shielded phases, and waters do not use amide. They have the amide, but most of the shielded phases are using this type of group, which is a carbamide group. And stable bonds and difunction silane, in the beginning, they touting advantages such as more stability at low pH, a wider pH stability, and less hydrolytic stability, less hydrolyzing. The bonding group is not as easily hydrolyzed as that of the monofunctional silane. And embedded polar groups, bond phases have different selectivity and generally give better peak shapes for basic analytes because also we have different selectivity due to mixed mode interactions. Here is some of the illustration on selectivity differences of the family of waters different bond phases over here. So here, and the nomenclature is kind of unique, and sometimes they might not disclose exactly what it means. So I try to help decipher some of these coding over here. So first one is called charge surface hybrid. We'll talk about charge surface hybrid later on. C18. So you have a elution profile like this 1, 2, 3, 4, 5, 6, 7. And this is a phenyl-hexyl group. It's a phenyl bond phase with a hexyl linker. Due to pi-pi-dipole interaction, you can see that there are some shifts of retention time, but basically give you the same elution profile. The fluorophenyl are quite different due to hydrogen bonding. You can see that things are, the elution profile are changed quite a bit substantially. Cyanol bond phase. PFP also give you very substantial changes. T3 actually, in my view, stands for trifunctional silane. T3, and often you see T3. The meaning is trifunctional silane. HSS stands for high strength silica, which is the traditional silica using monofunctional silane. And this is a stable bond of phases, which is not, there's no end capping in the bonding chemistry. Here's an application case study. This came from Brian Yang of Bristol Myers Squibb in the presentation at 2014 in New Orleans at PitCon, and showing here a charge surface hybrid using a phenyl-hexyl column, which is the one like this, and separation stability indicating method of a drug product with 2 APIs, and showing a pretty good separation of the retention marker cocktail using very simple mobile phases of TFA in acetonitrile and TFA in water. Another case study showing rapid method screening. This is the method screening of a falsely degraded sample of NCE. Often we do that to screen to see which is the bond phase the most effective in separating to basically key in on the best bond phase for our separation. And this is a difficult case because we know that C18 there's a compound that's hidden underneath the API peak due to the peak shape is kind of strange and kind of broad. So we do a screening with 3 columns. So a lot of time column screening, typically you can use 3, 4, up to 6 column in screening columns. Typically use a broad gradient, a very simple mobile phases to do that. And clearly in this case, the polar embedded phases is the best because it actually resolves 4 peaks rather than 3 peaks in C18. And phenyl is not a good choice. This slide demonstrates a case study called phase collapse or pore dewetting phenomena. It turned out that a lot of the column, when operating at very low percentage of methanol or acetonitrile, perhaps lower than 20%, under isocratic condition. And when you stop the pump, bad things happen because of a phenomenon called phase collapse, where suddenly you lost quite a bit of retention, uh, due to a phenomena that the C18 long chain hydrophobic phases actually collapse so that there's less uh, stationary phase available for retention, right? So that's not good. Uh, the the column actually can be restored, by going to a higher percentage of organic solvent to kind of wet the hydrophobic chain again, and you just cannot stop the pump at those conditions. Now, some of the polar embedded column, they will not face anything called phase collapse. So there are some additional research from Waters Corporation by Greedy and Tom Walter, and published on this phenomena. And they believe that rather than phase collapse, the phenomena should be better described as something called pore dewetting, shown in this schematic here. In this slide, we demonstrate a C18 bond phase designed for polar compounds. One of the weaknesses of reverse phase is the fact that a lot of time for very polar compound, there's no retention. And it's a problem with there's no retention, there's no resolution. So there's a bond phase developed about 20 years ago called the Waters Atlantis. And the strategy in the bonded phase design is instead of trying to bond fully, there's less bonding density. And also the bonded phase does not go through the end capping process. So to compare the conventional C18 column on the separation of some of the polar compounds, you can see that peak 1, 2, 3 is not much retained. The Atlantis under the same condition show much more retention, and therefore for separation such as polar drug, this is a little bit more desirable. On section 3, we go look into the Van Deemter model or the Van Deemter equation for band broadening versus flow. The equation was developed by Van Deemter, which is a Dutch chemical engineer in the 1950s, shown here in the reference. Now, in the Van Deemter equation, it was found that the relationship on something called a theoretical plate height, okay, related to the column efficiency is dependent on three different components, three terms called A, B, and C. Now, the reason why it's being called a plate or theoretical plate is the fact that for a chemical engineer, most of them were designing columns for petrochemical distillation, such as here. These are huge columns, at least 5 stories high, with different plates built inside to fractionate crude oil into different type of kerosene, heating oil, gasoline, aviation fluid, so on. So, it is a theory to relate the separation efficiency of the process with the chromatographic process. Now, obviously, in chromatography, you have many more plates, even though there's no physical plates inside a column. All you have are these porous particle column. So if you want to have a plate height which is small, so that you have more plates, more separation power per unit length. So there's three terms, A, B, and C, and they have different relationships with flow rate or linear velocity. So here is the plate height, and here is the linear flow velocity. A is a multipath term, which does not change in regards to flow, while B is something called the longitudinal diffusion, where the contribution of the B term are lower at high flow rate, but it become very high at low flow rate. Uh, Finally, you have something called C term, which is resistance to mass transfer, C term over here, which is very dependent on flow rate. And here is the Van Deemter equation, depending on some of the diffusivity of the analyte. U stands for the linear velocity, dp is particle size. So this slide is a schematic to illustrate the three Van Deemter's terms. A term is something called the eddy diffusion term, uh, which is shown here. It's kind of the multipath phenomena, right? For instance, we're injecting a small band, such as caffeine, and there's billions of molecules going through there. Each of these molecules or analyte would go through the column with a different path, such as a random walk. However, if you have a very tightly packed column with a very well-packed column, there's no voice in there. Ultimately, under isocratic condition, even though the bend is emerging broader, it should come as a pretty sharp peak, right? So this is the A term. B term is something called longitudinal diffusion. Okay, a lot of people might think that if you use low flow rate, you give the analyte more time to equilibrate, and therefore give you high efficiency. Actually, it's not true, because in the mobile phase, when the band is in the liquid in the mobile phase, if it's highly concentrated and the flow rate is too slow, then there's passive diffusion, which might broaden it. So definitely using the lowest flow rate is not the best. The best way is to use an optimum flow rate. Here is also C term, right? So one can imagine there's billions of particles of, let's say, a caffeine in this sample being flowed through the column, and they would go inside the pores and actually interacting with the stationary phase, such as C18 on that. So there's two profiles. There's a profile caffeine molecules, which is in the stationary phase, interacting with the stationary phase, most of the time inside the pores. And when they are interacting with stationary phase, they do not move. And here is the concentration profile of the caffeine molecule in the mobile phase, and they move. So it turned out that if the particle size is too big, the pore is, the diffusion length of the pore are substantial. There's a mass resistance to mass transfer in those cases. So if you have smaller particles, that would actually tend to reduce the resistance to mass transfer. So once again, this is the plate height, which is a composite of the three term, A term, B term, and C term. So here is actual Van Deemter curve, uh, uh, theoretical term versus an experimental plot. This actually slide is a courtesy of Neijun Wu of Celgene, okay, in which he actually did experiment on 3 particle size, 5 micron, 3.5 micron, and 1.7 micron. Okay, and with this Van Deemter equation, one can explain why smaller particle size or smaller dp is better for very fast LC for high throughput screening. Uh, definitely because of lower resistance to mass transfer, you can run it faster flow rate. Let's look at these Van Deemter curve. This is the composite terms from all the three terms. So for 5 micron, the minimum play height or the most efficient flow rate is right here. The optimum flow rate is here. The minimum play height is about 10. So it's roughly about twice that of the particle diameter, the minimum play height. For 1.7 micron particle, the minimum play height are much smaller, roughly about, once again, twice the particle size of about 3.5. And it turned out that the actual minimum play height is shifting to higher flow rate. So it makes the smaller particle column much more efficient at higher flow rate and faster separation, right? So one of the approximation is that the A term, the minimum A term, is equal to twice that of the particle size. The C term actually is proportional to dp squared. So the slope of the C term at 5 micron is substantially higher than that of the 1.7 micron, right? It's 3 times smaller particle. The C term is 9 times shallower. There's one way to estimate the number of plate in the column, and this is using the estimation, the quasi-equation of the number of plates equal to L, which is the length, divided by twice that of the particle size. So for a well-packed column, the minimum plate height is roughly equal to 2 times the particle size. Since n is equal to L over H, so one can calculate very easily what is n, right? So for a 15 centimeter column packed with 5 micron column, you go through the arithmetic, you have 15,000 plates. However, if you do the same thing for the 1.7 micron column or the sub 2 micron particle size, one only have to use 5 centimeter long column, and one can get basically the same efficiency, right? So that means you can use a short column, the column's at least 3 times faster, and you can get the same efficiency. This is a good thing to be. However, the problem with using smaller particle size is the fact that the delta P, or the pressure drop, is inversely proportional to dp squared. So meaning that when you go to smaller particle, the back pressure is substantially higher. This actually explains why we want to go to ultra high pressure LC system with higher pressure capability. So small particle is great for speed, but require much higher operating pressure. So here's my summary and conclusion. Bonding chemistry and residual cyanide. The traditional bonding chemistry uses the monofunctional silane. Residual cyanide reduces, the number are reduced using end capping procedures. Metallic oxide can activate residual cyanide group, causing peak tailing for basic analytes. Second one is novel bonding chemistry. Besides C18 bonding phases, other useful reverse phase bonding phases are phenyl, PFP, polar embedded phases for pharmaceutical analysis or other applications. The Van Deemter equation describes the theoretical plate height changes with flow rate or linear flow. There are 3 terms, A, B, and C terms. Here is the references. And here is the quiz with the answers here.